Hello and welcome to Perspectives on PH. I'm Steve Highsmith. PAH.TV brings you up-to-date coverage of the latest abstracts presented at the 2012 American Thoracic Society International Conference held May 18th to the 24th, 2012. We're pleased to have with us today Dr. Ioana Preston from Tufts Medical Center in Boston. During ATS, Dr. Preston chaired the poster session on pathophysiology of PH. Welcome, Dr. Preston.
Thanks, Steve. Glad to be here.
Doctor, can you talk about the importance of the posters that were presented and provide some highlights?
Sure. The poster session was very active. We had 25 posters with very interesting studies and findings that try to understand and detail the aspects of pulmonary hypertension with regards to pathophysiology. Among the posters, I would like to mention a couple of them that I thought they were novel in regards to pathophysiology. The first poster, which is called Autoimmunity and Pulmonary Arterial Hypertension, the Role of Leptin, was a study from France, from a research group in France, which looked at leptin in PAH. Leptin is a molecule that traditionally has been linked with obesity, but more so it has been shown recently that it promotes various autoimmune diseases by controlling certain white cells called T regulatory cells in their response to inflammation. So far, the role of leptin in PAH is unknown, and this group found that leptin serum levels are increased in patients with idiopathic PAH and in patients with PAH associated with scleroderma. And more so, the T cells from these patients who have a receptor for leptin have a reduced function in a leptin-dependent manner. And more so, when these researchers looked at the endothelial cells derived from patients with IPAH, they showed that these endothelial cells produce leptin at an increased level compared to controls. So taken together, these data suggest that leptin and possibly its receptor on the T cells may represent a new target for pulmonary arterial hypertension. So this is a new pathway that has been uncovered in PAH that warrants further study.
What are the conclusions then of this study?
It's too early to draw any firm conclusions, but targeting leptin levels to decrease their levels or enhancing maybe or restoring the function of the T cells may help in the treatment of pulmonary hypertension. And this is speculatory at this time, but it may prove to be a good target for treatment for PAH.
Well, thank you, Doctor. The second poster we'll be reviewing today is titled Pulmonary Hypertension Involves Augmented Smooth Muscle Gene Expression in Cells of Endothelial Genetic Lineage. Can you describe the objective of this study?
Yes, this is a very interesting work which came from the collaboration of a group from Stanford University and Sichuan University School of Medicine. And they looked at the plexiform lesions from the lungs of animals with experimental pulmonary hypertension. And although this is not a human study, I think it's absolutely important to understand how these plexiform lesions develop, what type of cells are in preponderance in these lesions, in order for us to find specific targets to improve these lesions. So this group looked at a model of pulmonary hypertension obtained by treating rats with monocrotaline and pneumonectomy. And after this treatment, rats developed very severe pulmonary arterial hypertension with plexiform lesions that resemble those lesions from patients with PAH. And they looked at the type of the cells that form these plexiform lesions. What they found was that the majority of cells are of endothelial origin, but they do not behave like endothelial cells, but they have markers of smooth muscle cells. So they undergo a transformation from endothelial cells to smooth muscle cells that make these plexiform lesions characterized by a mix of population.
So, Dr. Preston, what is the importance of this study?
I think this is very important in understanding what makes the pathological lesions in the lungs of patients with pulmonary arterial hypertension. And hopefully by understanding what types of cells develop and their behavior from these plexiform lesions will give us a better target to how to treat these lesions to improve the flow through these abnormal vessels in the lung vasculature.
Thank you, Doctor. Finally, there was an intriguing study looking at the cell-penetrating homing peptide CAR, which may enhance the delivery of PAH drugs. Dr. Preston, what are the clinical implications of this study, and how might this benefit patients in the future?
So this is another example of a study which shows wonderful collaboration between several research groups. And this is very novel. This study looked at a model of pulmonary hypertension in which rats are exposed to a molecule called sugen, and then they are exposed to chronic hypoxia, and they develop severe pulmonary arterial hypertension with plexiform lesions and very, very abnormal pulmonary vasculature. These researchers found a molecule, a peptide called CAR, C-A-R, which homes to the neovasculature of wounded tissue. And they hypothesized that by using this homing peptide will be homed into the blood vessels that are abnormal in the pulmonary vasculature. The role of this peptide is to transport drugs and deliver them to specific sites. So in this particular study, the homing peptide CAR selectively transported two drugs that have antihypertensive effects on the pulmonary vasculature, a rho kinase inhibitor called fasudil and a tyrosine kinase inhibitor called imatinib, who has been already studied in patients with pulmonary hypertension. The effects of these two drugs were enhanced when co-administered with a homing peptide, suggesting that this homing peptide delivers more drug to the wounded vessels in the pulmonary vasculature. What was also very important was that the drugs were not delivered in higher concentration in other organs. So with this type of enhanced delivery of PAH drugs, one would argue that we can deliver them directly to the vascular tissue in the pulmonary arteries, and yet avoid the side effects that these drugs can be associated with.
Thank you, Dr. Preston, for your insights into these important presentations from ATS.
Thank you for inviting me.
That was Dr. Ioana Preston from Tufts Medical Center in Boston. Thank you to all of the experts who contributed to this edition of Perspectives on PH. We look forward to seeing you here at PAH.TV as we cover the latest news in pulmonary hypertension.